Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Spatiotemporal Dynamics of Calcium and Neurotransmitters in Awake Mouse Models of Epilepsy. I'm Andrew Davies from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Neurotar, so a big thank you to them for helping to make this event possible. We're very fortunate to be joined by Dr. Vincent Magloire, Wellcome Senior Research Fellow in the Department of Clinical and Experimental Epilepsy at the Institute of Neurology at University College London, and Dr. Ken Berglund, Assistant Professor of Neurosurgery in the School of Medicine at Emory University. Today they will be discussing their research on the spatiotemporal dynamics of neurotransmitter and calcium imaging during seizure evolution in awake, head-fixed mice and models of epilepsy. Before getting started, I'd like to cover a few brief housekeeping notes to help you get the most out of your experience. First, this webinar is being recorded and will be available to view on demand shortly after the live event. Next, if you take a look at the interactivity panel on the right-hand side of your screen, there are 4 tabs corresponding to chat, Q&A, polls, and handouts. If you experience any technical issues, please first try refreshing your browser, as this fixes most problems. But if you're still having trouble, please reach out using the private chat function, and we'll help you to troubleshoot. We'd also love to hear from you, so please send any thoughts, questions, and comments using the Q&A tab, and we'll address as many of them we can in a Q&A session after the presentations. And finally, you can also explore relevant resources in the Handouts tab to learn more about some of the content, methods, and equipment presented today. And just before I bring on our first speaker, we have 2 brief audience polls for you. Our first question is, which of the following research methods do you currently use? Please select all that apply, and the options are 2-photon imaging, electrophysiological recordings, behavioral assays, optogenetics, functional ultrasound imaging, or wide field imaging. And again, please select all that apply, and I'll just give everyone a moment to submit the responses. And these polls are also available in the polls tab if you'd like to do them later. And thanks very much, everyone. And our second and final audience poll for today is, which statement best describes your situation? Here the options are: I currently work with awake rodents and am interested in head fixation techniques. I will work with awake mice in the future using optical or functional ultrasound imaging. I will work with awake mice in the future with electrophysiological studies, or I just have a general interest in the subject and technique. And again, I'll just give everyone a moment to submit the response. All right, wonderful. And thank you all very much. It is now my pleasure to welcome our first speaker, Vincent. Let's bring Vincent on webcam. Hi, Vincent. Welcome. Please take it away whenever you're ready.
Hi, thank you very much, Andrew, for the introductions, and thank you for having us today to speak about spatial temporal dynamic of calcium and neurotransmitter in epilepsy. So my talk will be more about GABA and glutamate imaging at the transition to seizures, and that's done at UCL, or it has been done at UCL. So before starting that, I would just like to briefly introduce more what is epilepsy. And epilepsy is around 1% of the population worldwide that is affected by this neurological disorder. In the UK, it's around 600,000 persons. And the origin could be various, from genetic brain traumas, infections, or abnormal development, for example. And what is important to remember is that it's around 30% of the patients are still pharmacoresistant in this disorder. And so we are working toward understanding a bit better this disorder to find new therapeutic targets. And so what I'm interested in this context is, why does the brain doesn't seize all the time, if you want. This is just a curve showing you that the incidence of epileptic patients in function of their age. And you see that at childhood and elderly, this is where you have more chance to develop an epilepsy. And that's certainly not necessarily or random, if you want, because during the childhood, your excitatory and inhibitory system in the brain is developing, and there is much less, or there is much more excitations than inhibitions. And during the elderly phase, it's a bit more complicated. We don't really know why, but we could assume that during neurodegenerations potentially, and we know that some part of the inhibitory system is also failing in this case. And so I'm studying sort of these questions in the lens of the inhibitory system, right? So we know that inhibition is crucial to keep excitation in check in the brain, and we also and because when we use like antagonists for this inhibitory system, we can recreate overexcitation and epilepsy in animal models, and we also treat epileptic patients with GABA agonists, for example, GABAergic agonist. And so the problem is that when you look at just in very simply like this, you can see, okay, there is like less inhibition in the brain, so let's put more inhibitions, and then we restore this sort of like equilibrium and we treat epilepsy. But the reality is that this inhibitory system is very complex, and there is like different GABAergic transmission that could be like phasic or tonic inhibitions, fast or slow inhibitions led by different type of GABA receptors. There is also the GABA B receptors that are like more metabotropic and that are pre and post-synaptic or acting on pre and post-synaptic side. And there is different type of transporter that reuptake the GABA from the extracellular space from the neuron and astrocyte. And when you think about this complexity, you then add the number of interneurons that exist in this environment, and you have like more than 20 different type of interneurons that has been sort of like characterized in the neocortex, for example. So when you start to look at the system like this, it's much more than just restoring inhibition. You need to restore the right inhibition if you want. And so that's what we have been studying for like a few years now at UCL, and that's sort of the question that we are asking: which part of the inhibitory system is necessary and sufficient to maintain physiological network stability? Do all interneuron class would participate equally to this, to restrain the excitation in time and space, and whether there is like global structures that would also participate to that. And so in this general context, what I'm going to present to you today is more about the inhibitory restraint. So what is the inhibitory restraint? Is like when you have a small focal point in the neocortex that is overexcited, like a number of pyramidal neurons that are overexcited. Generally, you have like what we call like a halo, or that's what we think. There is a halo of inhibitions that surround these excitations, at least initially, like you can see on the left panel here. And what we see happen during seizures, like so that means that you have like a very small excitation focus. But after a while, this small excitation focus, or the inhibition surrounding this small excitation that try to restrain these excitations, start to fail. Fail to do his job to constrain these excitations, and though the excitation take over larger territory in the neocortex circuit, and then etc. until you have like generalization of the seizure. And so that's what we are studying here, and we are studying at two different levels. At a subcellular level, we're studying the spatiotemporal distribution of neurotransmitter during this short period of short excitations to seizures, and also with different interneurons. And today I'm going to speak about the first part, the subcellular level. And this has been this is work that has been done in collaboration with Yoshiteru Shimoda, Mark Leite, Rob Graham, and Dimitri Kuhlman, at least as the main actors. And so how do we do that? So we use two-photon imaging, so two-photon microscopy, combined with optical sensors, genetically encoded optical sensors, that are going to sense the glutamate or the GABA from the extracellular space. So we have those proteins that we're going to fluoresce upon binding of glutamate or GABA. And so we express them like via like an adeno-associated virus vectors in the neocortex, in the visual cortex to be more precise. And 3 weeks later, we then perform a craniotomy above the region of interest. And we put like cover slip, we create a window of imaging if you want, and we use this ATFIX system that from the neurotransmitter system. And then from that, we have like on top of the imaging window, we also have like a wire. So we have also like an electrode close by the window, the imaging window, and we also have a site where we can inject a drug. And why we want to inject a drug is to create our epileptic models that's going to be via using either like GABA A antagonist or cholinergic agonist. So what we do is that we put the animal under this. So we inject the drug, and then we put the animal under the microscope, and then we wait for the epileptic activity to appear. And so that's what you can see. So here on the bottom part is the electrophysical trace of that is generated by our model. So this is, in this case here, we inject a bolus of picrotoxin 1 millimeter away from the imaging window, and so after 10 to 20 minutes you start to see those spikes, those intellectuals, what we decided to call them intellectual spikes. That's very sharp, short activity that can evolve, or that often evolve to like short seizures that are a bit more complex. And so during those activity, what we do is then we looked at region of interest that have expressed our GABA or glutamate sensors, and then we apply our spiral scan imaging to start to image either the GABA signals or the glutamate signals at the same time as the electrophysical signals. And that's what you could see here as a result. This is the average of all those line scans. So using this method, we can then look at the difference between the GABA and the glutamate transients during those activity. And what we see here, for example, is at the top on the red signals is your glutamate sniffer signals coupled with the electrocorticogram. And what you see is that you have a sharp increase of the glutamate during this very sharp, short event, which is also true for the GABA. The difference is that the GABA signals is lasting much longer. And so that's sort of like made by this decay tau here. And so from that, we can interpret that both are, you have a surge of GABA and glutamate during those activity, but the GABA is outlasting the glutamate during this intellectual spikes. And so when we start to look at spatially what is happening is that we can go closer to the focus than 1 millimeters, but from 1 millimeters to 2 millimeters away from the focus, what you can see is that the glutamate signals is maximum or high close to the focus, close to where we inject the drug, and drop down to almost nothing after 2 millimeters. And interestingly, that's the inverse for the GABA. You have like less GABA signals close to the focus and more GABA signals away from the focus. And that's sort of like summarized here. You have like a monotonic decrease of the glutamate in function of how far you are from the focus. So the closer to the focus, more glutamate you have, and further away from the focus, less glutamate you have. And for the GABA, it's much less obvious. It's more like potentially a bell curve. I mean, it's more like flat. And then so you have like a lot of GABA that stays around 1.5 millimeters away from the focus before dropping down. And so what we can interpret that, how we can interpret that as is that the GABA surround this hyperacceptable focus during intellectual spikes. And another aspect that we are looking at is that the propagation directions and speed of those signals. And so for the glutamate, you see that the glutamate is going away from the focus. So you can see it here as this is the average vector here. While the GABA, interestingly, or like it's more intriguing even, seems to go toward the focus. And the speed of propagation of those signals, so it's around like 60 to 75 millimeters per second for the glutamate, while it's like 3 times slower or at least 2 times slower for the GABA. And so the glutamate is close to what we've seen with like local field potential propagations in other studies. And so here it's sort of like what we see is that, and that's sort of intriguing, and we still don't understand exactly why it's like this, but the GABA and the glutamate waves are going in opposite directions and are constraining, or at least the GABA seems to constrain the excitation in this respect. So that's sort of the first observation, and that's what's interesting with this method is that that's the direct observation of the inhibitory response, and I think that's sort of the first way of visualizing it spatially. And so what we have seen is that the GABA is released during intellectual activity, that it outlasts the glutamate, and that it surrounds this hyperacceptable focus that we have created. And that's sort of like a cartoon that would resume what we have seen here. more interesting is that this method allows us now to truly study the transition from this very short interictal spike that is very local to more like the propagation of seizures. And the question is like, often what you see is that interictal spikes and the beginning of a seizure that we call sentinel spikes are very similar or are the same when you look at in terms of electrophysical signatures. But the reality is that with these methods and glutamate and GABA imaging, we could unreveal new aspects of these transitions. And so what we have seen in the past from other collaborators at UCL is that when you look at the calcium activity during interictal spikes or seizures, you see that there's some sort of a spatial continuum. So you have like an interictal spike. So this fluorescent here is like a calcium fluorescence, sort of a proxy for neuronal activity. And here they inject the drug. And so what you see is that interictal spikes, you have this focus of excitation that it's here, that's then fade away. But during seizures, at seizure onset, this sort of activity then continue. And it's really the same activity. And what we have seen is that around 1.5 milliliters away from the injection of the drug, that's where the seizures start. And so we use this information as well as our imaging to try to understand whether there is a failure of the inhibitory system at seizure onset and can we image it. And so for that, we repeat this experiment, but then we looked at specifically at 1.5 to 2 milliliters away from the focus, and we look at the GABA and the glutamate signals at the transition to seizures. And so what we've done here is that we looked at, if you want, 3 interictal spikes before a seizure happened, or a small seizure happened, and we look at the GABA and the glutamate signals this way. And what we see is that at least 3 interictal spikes before, you can start to see a gradual decrease of the GABA signals before seizure. And that's like mirrored by an increase of the glutamate signals 3 interictal spikes before. And so what those data suggest is that there's effectively a failure of the inhibitory restraint at seizure onset, and that the GABA imaging help us to reveal that. And so the questions now is like, is there like, or what type of inhibition, if you want, is failing? Is it like the dendritic inhibition, the somatic inhibitions? It's not yet clear. And interestingly, or more interestingly, is that here we've seen that we have been like few seconds before a seizure onset, we can start to have like a change in the concentration of glutamate and GABA in the neocortex, close to the focus. And so can the GABA and glutamate as predictive values for seizure onset that we could be then used to predict future seizures? And so on this, I would like to conclude that what we've seen is that the GABA surround an excitatory focus during interictal discharge, that the GABA and glutamate travel in opposite directions, and that's a very interesting and intriguing phenomenon that we are trying to understand now, and that we've seen a failure of GABA release at seizure onset. And these bring a lot of different questions, some of them that we are now studying, the mechanism of GABA release failure, which type of interneuron fails, and also how the inhibitory resistance evolve over time, but longer than just a few seconds, and can GABA and glutamate levels can be used to predict seizure in the future. And so on that, I would like just to thank people that have worked on this project, Yoshiteru Shimoda, Marco Leite, and Rob Graham, as well as Dimitri, that have been the main contributor to this work, as well as Jonathan and Lauren Luger from Janina Farm for the glutamate and GABA sniffer. And to finish, I would like to thank the founders that found these works, and also bring to your attention like something a bit different, but it's a preprint peer review platform that I will invite you to look at. Thank you for your attention, and I think on that I should give the hand to Ken, that's going to speak about calcium imaging in epilepsy, but also during spring depolarizations. Thank you.
Hello, everyone. Hi from Atlanta, Georgia. Thank you very much for joining my talk today. I am very excited to share some recent results from our lab. I'm also feel a little bit anxious because those almost all the data I show you today is yet to be published. So I appreciate any feedback. Now, our lab utilized several epilepsy models in rodents, and here's one example of EEG recording during one of the seizure models we use. It's a systemic injection of pentylene tetrazole, or PTZ. In today's talk, I exclusively use this generalized seizure model induced by this chemical convulsant. EEG was recorded in the mouse primary motor cortex, as you can appreciate in this representative recordings. Seizure consisted of several phases. Preictal spikes is a representation of pathological population spiking of neurons, which evolved into full-blown seizure, as the Vincent talk showed. So after somehow the seizure ends and the EEG goes almost flat, which is the this post-ictal quiescent period. Now today's talk is almost focuses on this post-ictal phase. You may think it's kind of boring because there's not much going on in terms of EEG, but I tell you, in terms of calcium, there's lots going on, and I hope you will enjoy my talk. Before I go into any technical detail, I just want to show you one representative imaging session during this PTZ model seizure. Again, so the yellow calcium indicator is expressed in neurons in layer, and we are imaging layer 2, 3 in the motor cortex. Now the preictal spike shows up those flashes, and you can also see occasional spike in the median neurons, and then seizure goes into the fringing field. And now you see what happens at the end of the seizure. There's this slow propagation wave going from left to the right, and the cytosolic calcium is actually elevated for a while, and eventually it goes back to the baseline, and then go back to the normal. Let's look at the data in a bit different way. So we have done this calcium imaging in a head fixed, but otherwise freely moving awake mice on the neurotransmitter floating platform. Neuronal calcium was imaged through a glass window in the skull. And these wires are for EEG recording. Calcium was measured in the population of neurons in the field, as well as in the individual cells, as shown in the left plot. These calcium transient reflects the interictal spikes, preictal spikes. During the seizure, EEG went crazy, and then there's an elevated and sustained calcium increase was observed. The seizure was, this calcium increase was followed by a second calcium increase. In the raster plot, you might be able to appreciate that there's actually calcium is propagating slowly. Now, this phase of secondary calcium increase is like antithesis of calcium imaging, because we use calcium indicator as a proxy of neuronal firing, when the cell depolarized the calcium influx, and we can see it as calcium. But this second calcium change doesn't have any corresponding neuronal activity. So what's going on here? Well, what's going on is a cortical spreading depolarization, or CSD. CSD is nothing new and has been studied for quite some time. CSD is a shift of 10 millivolt or so in extracellular DC potential, which slowly propagates across the neocortex and suppresses neuronal activity. It's often studied in the context of migraine and stroke. But the very first description in the literature was related to epilepsy. CST was discovered accidentally while trying to evoke a seizure in an animal model. CST happens in human as well. Here are some example of where CST was recorded through low-pass filter EEG in epileptic patients. Now we can study the slowly propagating calcium during CST, which has a distinct calcium signaling. We need some method to look at the calcium in a very different way than the ordinary calcium increase during the normal activity. Now I'd like to turn your attention to a calcium indicator protein for endoplasmic reticulum, or ER. So we decided to study CST with an ER calcium sensor, our charger, developed by our collaborator, Jenny Yang at Georgia State University. Our charger has a very unique calcium sensing mechanism. Unlike other conventional gekies with EF hand motif, it doesn't have any moving part, and instead, calcium bind site was engineered on the surface of the MAPO, a red fluorescent protein. Calcium binding directly affects the protonation state of the chromophore, shown in blue inside this beta barrel, and increase fluorescence. Because of this, fluorescence change is very fast compared to other geki that requires conformation change. Binding is selective to calcium, and its low affinity is ideal for sensing calcium in ER, as the calcium level is relatively high in ER compared to the cytosol. To keep the sensor inside ER, our charger has two ER retention motifs at both N and C terminals. We package it together with a yellow calcium indicator, XCamp-Y, developed by Haruhiko Bito of Tokyo University. Now, here's the reason why we are using yellow but not green. There's a substantial overlap in excitation spectra with our charger due to a modest redshift of the yellow geki. A single wavelength can excite both yellow and red during the two-photon imaging. We produced the adeno-associated viral vector and inject it into the mouse motor cortex. The idea is that we measure calcium in two different colors, in two different compartments, yellow in cytosol and red in the ER. The first step was to confirm our charger expression in ER in the mouse cortex after AAV injection. For that, we used immunostaining against SRCA2 calcium pump. SRCA2 is universally expressed in non-neuronal cells as well, but this methodological analysis confirmed co-localization of our charger and the ER marker. Then we performed craniotomy. Now we are looking into the brain from the above, as opposed to the side views in the previous slide. Our charger signal was actually pretty robust in vivo. Individual cells were really easily distinguishable in two-photon imaging in an awake mouse. Here's some calcium change during normal behavior. As we express the calcium indicators in the motor cortex, some cells showed calcium transient in the cytosol that coincides with locomotor activity during, just so you know, the neurotransmitter platform can track locomotion as well, like here shown in black. ER calcium stayed relatively flat during this period, and we saw no noticeable change in ER calcium either when the calcium traces were aligned at the peak of the calcium transient in the cytosol. Now let's go back to CST. The gold standard for CST is DC recording through a glass electrode and the DC amplifier. For that, we modify the cranial window so that it can have an access port. A tiny hole was drilled on the glass cover slip and filled with a silicon plug. Through the access port, a glass electrode can be inserted multiple times acutely. Here are some pictures with the glass electrode with wires for reference and EEG recordings. Now, one thing I want to add is that this convoluted experiment requires absolute stability and rigidity, as PTZ-induced seizures sometimes can go into a very violent conversion. the precision machine neurotransmitter head plate and holder has been very helpful in that regard. Here, the representative recording of a simultaneous EEG, DC, cytosolic calcium, and ER calcium. During the pre-ictal splashes, there's not much going on in the architecture. Now we need to wait until seizure embeds into the field. Now there's a sustained calcium increase in the green, but again, there's not much going in the red. But here, let's wait until what happens when the CST embeds into the field. You can see the increase in the green and the decrease in the red. And eventually, the ER calcium recovers, and then cytosolic calcium goes back to the baseline. Now, the period of DC potential shift, calcium increase and ER calcium decrease coincide pretty well. You may wonder if they are just a mere coincidence. Well, that they are not, as shown in the next slide. The PTZ model of epilepsy produces highly variable outcomes. All the recordings so far I presented had CST after seizure, but sometimes PTZ doesn't go above the seizure threshold and instead produce pre-ictal spikes only, as shown here. Yet another example is seizure without CST. When there was a seizure, or when there was no seizure, or there was a seizure but with a CST, in DC recording after seizure, there was no change in the cytosolic or ER calcium either. Here's the summary. The magnitude of calcium change is plotted in 3 different types of response after PZT injection: pre-ictal spikes only, seizure without CST, and seizure with CST. As you can see, the significant change in ER calcium was only observed during CST. More importantly, there was no calcium change in the same period of the post-ictal phase when there was no CST. These analyses confirm that depletion of ER calcium only happens during CST. One thing I'd like to point out is that although calcium increase in the cytosol is comparable between seizures and CST, depletion of ER calcium was only observed during CST, indicating that a large calcium increase by itself cannot trigger calcium depletion of ER. There's something else during CST. Now we looked into calcium response in individual cells. Here's another example of EEG, DC, and calcium traces in 4 different cells. We determined the precise timing of calcium change at the onset and then offset of CST, shown in light and dark blue ticks, respectively. We repeated this in 200 or so cells in the field of view and plotted the relative timing of calcium change in the color-coded manner. I hope you can appreciate that calcium propagates from left to right in both green and red channels. Then we applied 2-dimensional linear regression to calculate propagation calcium wave as vectors, which represent both direction and speed. This method is detailed in our recent publication. On average, CST propagated at the speed of about 70 microns per second in both channels, which is consistent with the literature. The offset of CST also can be seen as propagating waves, but the speed was much slower. They propagated in almost the same direction. During the duration of seizure was about a minute or so when determined in both green and red channels. All in all, calcium waves traveled in almost identical ways in the cytosol and ER. Here's another way to look at the propagating waves. Individual cells are positioned along the propagation vector. The linear regression represents the speed of the propagation. Steeper is slower. What you would notice is that the red dots are shifted to the right compared to green dots, indicating that ER calcium wave is trailing cytosolic calcium wave. This is also true for the offset of the CST. On average, there's about 1 second or so delay of ER calcium wave relative to cytosolic calcium wave. We interpret this as during CST, the initial influx of calcium into the cytosol triggers further calcium release from the internal calcium store of ER. Depletion of ER calcium during CST may be caused by calcium-induced calcium release. So what's the implication of CST in the seizure physiology? The data I have shown today came from the PTZ injection, but CST has been reported in wide variety of seizure models in rodents. Here are some examples from other labs in both acute models as well as spontaneous seizure after a kinase injection. As I explained at the beginning, CST is often studied in terms of migraine and stroke, where negative consequences have been reported. However, in case of seizures, CST always appears at the end of seizure as if it signals to terminate seizure. CST can be induced on demand by various stimulations, such as application of high potassium. And in fact, such treatment has shown to stop ongoing seizure. That CST may be an innate protective mechanism in the brain to stop seizures. We would like to further elucidate the role of CST in seizure physiology. To that end, we implemented a way to evoke CST reliably other than PTZ-induced seizure. For that, we used the electrical stimulation. We implanted another pair of electrodes and evoked CST and applied high-frequency stimulation. When high-frequency stimulation applied, we observed CST. This is going to be my last video. So we applied the high-frequency stimulation for 10 seconds or so, which evokes CST, and you can see the propagating calcium waves in both green and red channels. And just like PTZ in CTZ, it will be recovered after a minute or so. So here's a summary. Electrical evoked CST appears to share many features with the ones after PTZ-induced seizures. The two waves propagate about the same speed, if slightly faster than the CST after seizure. There was also the 1 second lag between cytosolic calcium and ER calcium. Thus, depletion of ER calcium is a conserved feature of CST and not only after the ones observed after seizure. Here's a summary. So we developed a method to monitor ER calcium, cytosolic calcium, EEG, and DC potential at the same time. CST was observed at the termination of seizure induced by PTZ. This sustained decrease in ER was only observed during this terminal CST. ER calcium was mostly stable otherwise. The decrease of ER calcium had a little bit of delay compared to the cytosolic calcium, indicating calcium-induced calcium release. Here's my acknowledgement slide. So the whole project was pretty much single-handedly completed by a very talented MD PhD student in the lab, Matthew Stern. Here's a picture when we visited Japan last summer. This is a collaboration of, so the agriculture development is a collaboration with Jenny Yang in Georgia State University and also the funding. Thank you very much.
Thank you, Ken and Vincent, for those fantastic presentations. Just a reminder, while we give our speakers a moment to grab a drink and catch their breath before the Q&A, if you have a question you'd like to share with them, you can submit it using the Q&A tab on the right-hand side of your screen. And if we don't have time to cover any of your questions today, we'll certainly send all of them to our speakers, and we'll release a Q&A report within a few weeks, so please stay tuned for that. I'll also take a moment to launch our survey shortly, which you can complete at any time, and also following the webinar. We do read all of your responses and really appreciate your feedback. And with that, it is now my pleasure to welcome Ken and Vincent back. Welcome back both, and thanks again for those great presentations. Ready for some questions?
Sure.
Perfect. Maybe we'll start with Ken. Following on your conclusions there, there's one here. Do you have any evidence that the depletion of the ER calcium is, directly mediated by the calcium-induced calcium release?
Yes. So we try to block the CICR by pharmacology. Now we have the access port to deliver drug into the brain, but the result was somewhat inconsistent. So instead, what we are looking for, we're looking for another approach to study this, which is the brain slice preparation. CST has been, studied and can be involved in the brain slice preparation as well as by either hypotassium or electrical stimulation. And the exposed brain is more easy sample to do pharmacology. So that's something we are planning to do.
Maybe a quick follow-on to that for both of you is, obviously there's a huge advantage to doing the in vivo quantification. Are there any methods to validate the results that you're seeing that are easily done? I suppose that's the $1 million question part of it, isn't it? The easily done part.
Easily done? I don't think so. I guess we moved toward in vivo as well, because a lot of what we have been doing in vivo has been, if not shown with other methods in vitro and ex vivo, have been modeled, or at least the concept was already there. So it's sort of, we're trying to move toward in vivo to see whether there is more, like if it's holding toward like closer to human in a way.
Right.
But is there a way to make it easier to validate it? And I don't know, I guess modeling is one way to think about it. Trying to model part of what we see in terms of dynamic with simple mean field model. We've tried that a bit, and so the data are going to, it's helping to find some sort of mechanism this way. But is there an easy experimental approach?
So few experiments are easy, I suppose. That makes a lot of sense. This is the next evolution to show the natural state, right, rather than the in vitro experiments. Yeah, that's fantastic. A follow-on.
May I ask you questions as well? Do you mind?
Of course, please. Yes.
Yeah. All right. Thanks, Ken. Thanks. It was it's very, very intriguing. Spring depolarization are very intriguing as well. And I think one question is so with your system with the cytosol versus the ER. Calcium imaging, I was wondering, when you see during the spring depolarizations, what's if I understood correctly, initially, from the speed of propagation from cell to cells, is it correct? You see that the calcium, cytosolic calcium, seems to be the first one, and then the ER depletions arrive, right?
That's correct.
But then, after this initial period, that I don't know how many seconds it is or how many milliseconds it is, then could it be the inverse, right? The calcium that you see, or the calcium activity that you see in the cytosol, could come from the ER alone. Is it correct?
That's right. So
how much you know that it's not neuronal activity, or is there a way to understand whether neuronal activity contribute to the end of this spreading depolarization, or is only the calcium from the ER? That's sort of the question.
Yes, that's a great question. So I was very puzzled about this finding because the thing I was shown in terms of ER calcium is that, release of calcium from the ER, and eventually, ER is going to pick up the calcium from the side store or from the extra space eventually. But that kind of coincides with the change in the DC potential. And then none of the, the calcium, ER calcium regulation is primarily dependent on the voltage, the membrane potential. So we really want to understand why, the global change in the membrane potential affects the physiology of ER calcium. That's some like an open question.
Cool. Thanks.
Yeah, fantastic. Maybe one for you here, Vincent. Obviously, all beautiful results, both as well. The GABA. What could be the potential source of the GABA that you're seeing?
Yes. So in our case, what we image is very superficial. So it's like 100 microns below the surface. So it's going to be layer one, what we call layer one of the neocortex, and there the main so there's like different type of interneurons there, 4 or 5 type, and also like some axon coming from like deeper layer from the somatostatin interneurons. So we have like a source of 5 different type of interneuron that could be that we are observing through this imaging, but to separate them all, it's the next challenge.
Right. Okay, great. This is a great question for both of you, really. So that you've obviously discussed specific models. To what extent and Ken touched on this as well. To what extent would you expect these results to carry over into other models of epilepsy? And perhaps even the clinical condition. You also both touched on that. Maybe we can start with Ken.
Yeah, so you see, I have some slides from a result from other labs. So it seems like CST is pretty universal among, all the seizure models tested. And so what we'd like to address in terms of, the relevance in the clinics is that, for example, the speed of propagation, we determined it's about 70 micron per second. And there are, a bunch of people has done EEG recording in the human patient that can be used to estimate the speed of propagation in the human patient. I wonder if those numbers matches or not.
Okay, great.
So that's some direction we want to go.
Right, fantastic. And Vincent?
Yeah. So I mean, what we've seen here is like mainly like one model of picrotoxin. We have done the same job with pilocarpine. It's another agonist, so it's similar system. It's an acute model, but the data are old, so they are the same. So that start to be reassuring. Now, if we speak about chronic models or like let's like more epileptic patients, then I think there is some evidence that there is more than I think there is evidence that there is an inhibitory restraint, and that has been measured mainly by electrophysiology in epileptic patients and also animal models, and that it collapses. So we know that as well. Now, how does it collapse? It's still a $1 million question in this case. Some recording in human patient would suggest that maybe like interneurons stop firing. At least some interneurons could stop firing, but can we study that more in detail in patients? That's again very complicated, I guess.
Okay, excellent. Maybe continuing on that thread a little bit, are you aware of any existing therapeutics that act on these mechanisms specifically that you're describing, or is this more a question of identifying potential new targets?
Yeah, I guess it's so. Okay, I think we are more like on trying to identify new targets. Even though those mechanisms, there's one that's this sort of failure of GABA release that could be because of interneuron stop firing. Is sort of known since like 30 years, and others as well. Now to develop like a good target, first, as you say, we need to find evidence for that in vivo, and that's what we are trying to do. And people are working on trying to rescue this sort of abnormal behavior from interneuron now. Yeah. At least they are betting on that. Yeah.
All right. And any thoughts there as well, Ken?
Yeah, so at the end of my talk, I showed CSD can be evoked by electrical stimulation. Electrical stimulation has been shown, very, a promising effect in to treat epileptic patients through RNS or DBS. And then we look into the parameter for these stimulation. And sometimes, it's stimulation is kind of equivalent, like kind of high frequency stimulation, like 140 hertz for 5 seconds or so. So I wonder if some of the effect, we've seen with those stimulation in the human, electrical stimulation in the human patient is mediated by spreading depolarization or not.
Okay. Oh, wonderful. Maybe following along in that lines too, there's a question here. Is the spreading depolarization actually anti-epileptic? And importantly, I guess, is there a downside to spreading depolarization?
Sure. So, well, for the short answer is we don't know. But we noticed, so I showed you the PTZ doesn't necessarily produce CSD after seizure. Sometimes, the seizure just ends without CSD. And we noticed there are two kind of differences between seizures with and without CSD. One thing is that frequency of post-ictal spikes. So sometimes after seizure, you have the brain keeps producing those ictal spikes. And it seems like when we have CSD, they're less likely to have those kind of post-ictal activity. And then another thing we notice is that when the animal doesn't have CSD, and then when the animal keeps producing post-ictal spike, sometimes, oftentimes, animal have a second seizure, which never happens when we have CSD. And then when we have second seizure, again, in most cases, animal died. So it seems like CSD is really preventing animals some very harmful the effect of seizures.
Okay.
We are trying to quantify these numbers right now.
So is the suggestion then that during CSD you're in an effective refractory period almost for
exactly. Yeah, it's completely abolished any activity, like regular activity. Spiking activity. So it seems like that's like key effect of the CSD in epileptic seizures.
Okay, great answer. Maybe we'll go to a couple of sort of technical questions here. For Vincent, are you imaging GABA and glutamate transients at the synapse?
Right. We are no. We are not at the synapse alone. Let's put it this way. It's more like expressed everywhere on the membrane, and so the GABA and the glutamate that we sense is both extra or it's synaptic and extrasynaptic. Yeah. So we don't have like the resolution, or we don't go at microscale this way.
Right.
Yeah.
And then for Ken, one about the sensor you're using. Does the red ER calcium sensor show photo switching behavior? Can you combine it with other green fluorescent proteins, optogenetics, other labels, I suppose?
Yeah, so I get this question a lot. So to be clear, the photo switching it's pretty common in any fluorescent protein. So after extensive excitation, the molecule goes into non-excitable dark state. So but the thing is that when you apply short wavelength of light, the molecule becomes excitable again. So when you switch excitation light wavelengths, then you have this sudden jump of fluorescence, even though there's actually no change in the calcium or any underlying molecule. So that would be an issue if we want to. Now, animal apple is known to have photo switching pretty well. So the way we avoid that problem is to use the single wavelength of the light, so that we just keep using the same light to look at both yellow and red indicators. And we try several ways to induce artificial this photo switching by applying LED light or using a different wavelengths or shorter wavelength of two-photon laser. But we haven't seen any photo switching, perhaps because we are doing two-photon imaging, two-photon excitation with the eye catcher. But I kind of, if we use one photon excitation and then switch between blue and green excitation light, then most likely there's a photo switching.
Okay, great. There's a question on the registration form about optimizing parameters when doing these methods. Any other additional thoughts? I mean, you've touched on there, obviously. Any additional thoughts about the best way to do it? I mean, I suppose stability is paramount.
Yeah, so I think that, well, what's paramount is the clarity of the cranial window. So you have to keep under control, no bone regrowth, no the tissue scar forming. So that's absolutely important. In terms of two-photon imaging, we didn't see any downside, or even though we increased the laser intensity a lot. So the signal in both red and green channels is very stable over time. That's something we applied at the beginning, because for the seizure recording it takes tens of minutes to an hour, but the signal was absolutely stable. So we didn't have the issue of photo bleaching. So the only thing we are a little worried about, we need to take care of, is the saturation of the signal, because during the seizure there's a massive calcium increase, and the signal goes up, all the way up to the ceiling. So we want to make sure we can still, image during those periods by, just tighter down. The gain and the laser intensity.
I suppose
you were imaging for an hour continuously? Yeah. Wow. Okay.
So we want to know CST, whether or not CST happens.
Yeah.
And then sometimes it takes a very long time.
Yeah.
You have a very large dynamic range to capture, I suppose, right? With the spikes, yeah. Any additions there, Vincent? Any?
No, I think Ken covered most of it. Stability and clarity of the window is clearly
are there any,
on top of the rest.
Yeah, perfect. Are there any specific nuances to doing this type of imaging in epileptic models specifically? I think, Ken, you touched on it, that there can be some, significant movement.
Yeah, the movements. I think the neuropilus activity is going to be very different as well. And yeah, I guess you are dealing with. I don't know whether you do it acutely or chronically. I guess some people do that chronically as well. So you deal with an animal for a long period of time.
Mm-hmm. So right. So the convulsive seizure is nothing like regular walking. Animals convulse a lot, and I think we are a little fortunate that neurotransmitter stages really keep the imaging plan very stable. Sometimes we have a little a shift in the plan, most likely because of some fault in the during the surgery. Maybe the head plate is not firmly attached. But as long as we have nice head plate, and then we just insert the head plates firm and nicely into the holder, then the image I showed today is after motion registration, but most of the recording without motion registration, we can still see quite good recording. Okay,
fantastic. Well, we're almost out of time here, but maybe we can wrap up with a couple of questions here. One for Vincent. What is the possible mechanism for this inhibitory failure?
So I just touched upon it. I mean, the one that we are observing with this GABA imaging, it's presynaptic, right? Or at least if it's synaptic, it's presynaptic. It's from the presynapse, it's the release. And so there's few mechanisms that could be put together. One is synaptic depletion. Too much activity makes that there's no GABA transmitter at the presynapse, or it could be interneuron depolarization block. So the interneuron stop spiking, basically, because they are so depolarized that that they can't repolarize there. And the last one is presynaptic GABA B inhibition as well. But the thing is that those ones are the presynaptic GABA B and the synaptic depletions often happen at much faster scale. So it's more like 10 to 20 hertz, while we are dealing with something that's below 1 hertz in terms of activity. So we think that the main one would be interneuron depolarization block. This needs to be investigated or shown.
So watch this space, future studies. Huh?
Maybe.
Maybe. Okay, there was one question from the registration form. Maybe we can get to quickly. I'm not sure if you have an answer here, but do you know how high the intracellular sodium peaks during seizure activity or spreading depolarization? And then as a second part, are there other labels that can be used to study that in a similar fashion to what you've already done?
Yeah, so so we did measure sodium, so we didn't know. Another thing I should mention is that the DC recording I showed today is like population response of the extracellular space. So it's like 10 millivolt or so, as I said during my presentation. But we don't know exactly how millivolt depolarization each individual neurons are receiving. Nowadays, voltage sensor, the fluorescent indicator proteins is pretty good for the membrane potential as well. So I was wondering, maybe we should look into that direction to measure the membrane potential of individual cells, and we can do so at the same time looking at the ER calcium in red. That would give us more like a precise, timing as well as the amplitude of the spreading depolarization.
Okay, excellent. Maybe just to wrap up, I suppose is it possible that there is a therapeutic combination here where you rescue the inhibition, before seizure, and then augment perhaps the cortical spreading depolarization afterwards? As a sort of complementary treatment? Is that a possibility?
Well, I mean, I think, it depends how you see the spreading depolarization. If they are a consequence of seizure, or if they are like independent of seizure, or if they are like some sort of intermediate. But yeah, maybe you could think of something around that. I don't know what you think, Ken, but I guess that if you see it as an anti-epileptic, or that spreading depolarization are a mechanism to shut down the entire circuit. I mean, you need the seizure first, then. But I don't know if it's always the case.
Yeah, so I was kind of interested in if there's any medication that can affect ER calcium. For example, staptogagin, it's a SACA2 blocker, and then inhibit the ER calcium regulation. And there's actually some publication using staptogagin, the drug, as anti-epileptic drug. So maybe there's some drug that acts on the ER calcium that have some anti-epileptic effect, and I'm very interested in study those.
Okay, wonderful. Unfortunately, I think that's all the time that we have for today. But I really wanted to thank both of you for sharing your thoughts and insights here today. It's been a real pleasure having you with us.
Thank you for having us.
Thank you.
Thanks very much, both. Also, a big thank you to our audience. We know you have very busy schedules, and we certainly appreciate you joining us today. If you haven't already, we now invite you to take a moment to share your feedback on our survey. We do read all of your comments and greatly appreciate your input. A huge thank you as well to Neurotar for making this event possible. If you do still have a question, it's not too late. Feel free to submit it via the survey. We'll forward it along to our speakers following the event. And if you're interested in learning more about the research presented today, we also invite you to check out the resources panel. In closing, thank you very much for joining us today. We hope you enjoyed this Inside Scientific webinar, and we hope to see you again next time. Thank you, everyone.